Everyone look up for a second. I don't want to affect your story, but I've gotten some good feedback from you guys, so let's do this. As I mentioned, a certain organelle, some of you are like, wait, how would that be involved? Remember I said, think outside the membrane? Yeah. So if this doesn't affect your story, and don't change your story if you're done. Okay? The reality of this little challenge is to see how you can talk about a eukaryotic cell and the division of labor for this one task, producing a secretion. You have cells in your body that do it all the time. If you take anatomy and physiology with me or Mr. Schweitzer, you're going to see we have tissues that are specialized in producing secretions or a product that is made in one part of the body that goes to another part. This is the feedback. As I was walking around, I pointed out, secretion, biosynthesis. In the back, someone mentioned how digestion would be involved. So let's kind of brainstorm. How digestion, would that be involved? All right, let's start at the beginning. In the beginning of this cell story, you're told, describe three processes, really. Describe the organelles involved in production, modification, and transportation. Right? Each one of those three things has got more than one organelle involved. Okay? So don't just raise your hand and yell out an answer. So let's see, Andrea, do you understand how chromatin would be involved? Would chromatin be involved in this process? Yeah. Yeah.
I'm so wrong.
Okay, now stop right there for a second. I just mentioned chromatin, and did anyone say chromatin? All right. Some said yes, some are like, wait, I didn't mention that. And some of you don't do this. I didn't mention that one. I didn't mention, oh my God, I'm going to, no. Think about the process. This is, yes, you're writing as a story, and I've been looking and seen some great stories. Some are doing it first person and stuff like that. Some you might even have someone read it. Read the story, and we can even put it up on the channel. Okay, so,
I would do it in a Russian accent.
so here's the deal. So see, chromatin, because we haven't learned about it yet, but we're going to learn DNA determines the ultimate amino acid sequence. What do amino acids actually make? Proteins. Okay? So if we go back, you've been given a job to produce a protein. So the machinery of the cell is going to kick in. All right? So I talked to a couple of you while we were walking around. So I want you to raise your hand and yell it out. Different organelles that you mentioned. So chromatin would be like, say, the nucleus saying, hey, let's make this protein. Specifically, the DNA is going to direct the production of a molecule called RNA, which we haven't learned yet, but we'll get there. And then that RNA is going to go to the site of protein production, the actual protein production site. Where would that be? Ribosomes. Okay, so let's think about it. We've mentioned chromatin. We've mentioned DNA and RNA, all taking place in the nucleus. And then outside of the nucleus, the ribosome, say, would it be on a free ribosome or an attached ribosome? An attached? Someone said attached? Okay, that means it's attached to what? What's our next organelle to mention? The endoplasmic reticulum. Alright? So let's follow a progression. Say it's right here. So we started, the message came here, and so ribosomes are producing this protein. What's going to happen next? Huh? It's going to, is it going to just fly through the cytoplasm by itself?
No, vesicle.
A vesicle. A little bit of the endoplasmic reticulum is going to surround it. It's a membrane. It's a membrane that separates it from the chemistry of the biochemistry of the cytoplasm that could alter or break that protein down. So it's protected. So now it bubbles over to the Golgi. Now, it bubbles over, and then it's going to get modified there, right? In the Golgi. So now we're talking about Golgi. There's one thing that's not on this picture that you can't see. Jackie, that's going to help that vesicle transport itself. What's that called? We talked about it. All right, the vesicle, does it just float over, or how does it actually move through the cytoplasm? You're covering your mouth with a phone, I couldn't help you say. Yeah, the vesicle. Well, does the vesicle just flow through the cytoplasm, or is it connect to, say, like a little cable?
Oh yeah.
Oh yeah, what? What's that called?
Oh,
Yes, say it loud.
Cytoplasm.
Cytoskeleton. So what you can't see here is there's like, imagine a whole bunch of like tubes who's like shooting out here, and the vesicle has a little pathway where it walks its way over to the Golgi, then it's modified. Okay.
Walking on sunshine.
Well, what's going to happen next? We've talked about production, we've talked about modification and transportation. Now we've got to do transportation outside the cell, right? So this little vesicle, imagine this going, and it's going to do a process, submembrane group. You should be able to tell us this. Remember the poster group? What's it called when something leaves the cell in a vesicle? Well, we're saying secretion, but is it endo or exocytosis?
Exo.
Exocytosis. And so that substance will be done here. Let's say this is a blood vessel, and now it's going to go to that spot. Now, there are other organelles that will be involved, so we're going to go back. All right? James?
Yes.
We were talking about one. What organelle and how would it be involved?
One that's involved.
Yeah. You talked about digestion earlier.
Wait.
Yeah. Okay. He just said the lysosome could be involved in this, and most of you right now might be thinking, why?
Why?
Why would what's the lysosome do?
It
Wait for it.
It like breaks down like poisonous substances and like digests stuff.
Okay. It doesn't like to it actually does do it. It breaks down poisonous substances, but it also digests. So let's go back to this. A protein is made up of what? Amino acids. So what a lysosome is going to do is it's going to break down on, say, some stored proteins. So you have individual amino acids so that you can assemble a new amino acid chain. So we just mentioned lysosome, but that means we should also mention another organelle now. Yes?
Peroxisome?
No, but it's something very similar. Well, actually, no. It's something that, where would the stuff be stored?
Vacuum.
Vacuole. So now we just added in these two into this whole urosecretory cell. The vacuole and the lysosome, they're going to break down the raw materials. So when the DNA says, hey, make this protein, the ribosome says, okay, here's the protein I'm going to make. Does that make sense? So we've kind of mentioned a whole bunch of organelles, almost everything in this picture. There's some we haven't mentioned. Two that we haven't mentioned that we could mention.
Mitochondria?
Mitochondria. Why would we mention?
Because it's powering everything to be able to do that.
Alright, so that would kick in and start, hey, we got to make a protein. We need some energy to do it. So then the mitochondria would kick in and produce some more ATP, the chemical energy. One last organelle we haven't mentioned that we could mention.
Nucleus.
What in the nucleus?
Nucleolus?
What does the nucleolus do?
Nothing.
It does what?
It like makes
It makes more ribosomes. So that could be involved. If we're saying, hey, we need to make proteins, well, maybe the nucleolus is going to trigger the production of some more ribosomes to make that protein that we're going to secrete and send out. Alright? So just one simple story of urosecretory cell could help you review and think about all the cell organelles that will be involved. The only one we haven't mentioned is the centriole.
That thing.
And it's a stretch, it's a leap of cell. Well, he got so tired from doing that, decided to divide, and then it could be involved in mitosis. Alright, so that is your cell story. We need to move on to the next thing, but go ahead and just, if you want to tweak yours, you're going to turn in your story at the end. Alright?